All right, in this video lecture, I am continuing on in chapter 14. So we just covered 14.1 RNA, where we talked about the structure of RNA and how it differs from DNA. So we're actually going to review some of that on the next slide here. So let's talk about some differences between RNA and DNA. Well, RNA, remember, is a single-stranded molecule where DNA is double-stranded. RNA is a short-term information molecule and DNA is long-term. Once RNA leaves that nucleus, it starts to degrade and be broken down by enzymes. Some other differences is that RNA has some different nucleotides, A, U, C, G. Okay, adenine, uracil, cytosine, guanine. DNA, A, T, G, C, right? Or CG, however you want to say it, but adenine, thymine, cytosine, guanine. And different sugars, okay? DNA stands for deoxyribose nucleic acid. RNA stands for ribose nucleic acid. What is RNA made up of? Same, three things. Sugar, in this case ribose, a phosphate group, and a nitrogen base, okay? So nucleotides. There's four different nucleotides, each for the same letter. What are the three main types of RNA? So we really kind of talked about messenger RNA in 14.1. How do we make messenger RNA through the process of transcription? But that's messenger, that's the first one. Second one is rRNA, which stands for ribosomal RNA. And ribosomal, well, makes up the ribosomes. And then tRNA for transfer RNA, they carry amino acids because the end game here is making proteins. And proteins is a macromolecule, okay, a big molecule for life, made up of tiny little subunits or monomers of amino acids. And so each tRNA will bring an amino acid, so those amino acids will form a bond, and we can create a chain of amino acids, also known as polypeptides. So in 14.1, we focused on messenger RNA, and in this video lecture, we will focus in on tRNA and rRNA. What is the process of going from DNA to RNA? That is called transcription. Transcription. What are some similarities between transcription and replication? I think there's like three of them that you can say that they both use enzymes. Transcription uses RNA polymerase. Replication was DNA polymerase. You can say like, a template is being used. Kind of like three basic steps where somehow DNA kind of gets opened up. And then you have nucleotides that match complementary. There's base pairing going on. A will pair with U. C with G in transcription, and replication it's A pairs with T, and C and G. So yeah. So today's learning objectives, I'm going to talk about the genetic code and then describe how ribosomes and tRNAs assist in assembling proteins and how it kind of ties back to genetics. So the genetic code is actually the nucleotide sequences of the mRNA. Okay, so how do we get that mRNA? Well, that was through transcription. Think of DNA inside the nucleus as like a big recipe book for every single type of cell possible. Skin cell, muscle cell, maybe even like different hair color, eye color, skin color, whatever. But the thing about the DNA is that it never leaves the nucleus. And where do we find ribosomes inside of a cell? We find them in the cytoplasm. So how do ribosomes get instructions from DNA to make proteins, to make things for the cell? The answer is mRNA, right? So mRNA is the messenger, okay? The base sequences is kind of the genetic code. This code is read as a triplet, okay? 1, 2, 3. 3 nucleotides on the mRNA will code for one amino acid. This code is called a codon. So this is one codon, this is another codon. And it is non-overlapping. So what that means is that you read 3, those 3 are done, and you move on to the next 3. You don't say, okay, I'm going to read these 3. Oh, now I'm going to read these 3. No, now I'm going to read these 3. That's overlapping. These are non-overlapping. So you read the first 3, and then you move on to the next 3. There are 64 possible codons that code for 20 different amino acids. And you might be wondering, how in the world did they figure that out? Well, they kind of used a little bit of math. Okay, they knew that there was at least 20 different amino acids in nature. Animals make some of the same amino acids that we do. Some of them can make more than we can, and that's why we have to eat. Because what we eat, when we eat protein, we're getting the amino acids that we need to, make proteins inside of us. So here's the math. We know that there are 4 nucleotides in RNA. Okay, A, U, C, G, right? So they said, okay, if each nucleotide coded for 1 amino acid, how many amino acids can I code for? Well, the answer is 4, right? 4 by itself is 4. So they said, okay, the code is not single. What if the code was a double, where 2 nucleotides coded for an amino acid? So 4 times 4 is 16. Okay, we're getting closer to 20, but the code is not a double. So they said, okay, what if the code was a triplet? 4 times 4 times 4, 64. Okay, and you can see that this number is bigger than 20. So that means that some of the codons, there are multiple codons that code for the same amino acid. So let's just take a look at cytosine, or no, let's look at cysteine. Okay, I said that wrong. How you work a codon wheel is you start in the center and you work your way out, because the code is a triplet. So this is the first letter, the second ring is the second letter, and the third ring is the third letter. U, G, U, that's 1 codon. U, G, C, that's 2 codons. So here we have 2 codons that code for cysteine. Leucine has a lot of different codons that code for it. Okay, let's just count here. 1, 2, 3. 1, 2, 3. There's 2. 1, 2, 3, 3. 1, 2, 3. There's 4. Okay, you see any other leucines? Oh, here we go. 1, 2, 3. That's 1. 1, 2, 3. That's 2. Any other leucines? Okay, so there are 6 different codons that code for leucine. Okay, so in your notes, I think you have some examples here. It says UUA. So UUA, hey, it's leucine. Now, if you ever get an assignment, you can just write the first 3 letters. Okay, but for the sake of this lecture, I'm going to write it all out. Leucine. UUG. UUG. Hey, another leucine. And I think I just did that to show you that, hey, this code is redundant. There are more than 1 codon that can code for the same amino acid. UGG. UGG. Tryptophan. You may have heard of that amino acid before. It's common in turkey. Supposedly it's an amino acid that makes you sleepy, but I've read some conflicting results or studies on that. ACG. ACG. Threonine. So you just write THR, or you can write it all out. Alright, so that's how you use a codon wheel. You're going to be using that a lot for an assignment here. Okay, so how does translation work? Well, first off, what is it? Well, we are going from RNA to protein. So we started this chapter talking about DNA going to RNA in the process of transcription. And now we're finishing it, RNA to the protein in the process of translation. Molecules involved, wow, that's supposed to say protein. Molecules involved all 3 types of mRNA or RNA. We have messenger RNA, our ribosomal RNA, which is going to form the ribosome, and then our transfer RNA, which is going to carry the amino acids. So I'm going to talk about my diagrams here because they're kind of weird, right? So this big green glob here, that's my ribosome. And you can see it's made up of 2 subunits, a large subunit and a small subunit. When they fit together, we get these chambers. 1 fold chamber, 2 fold chambers, and this one's a half. This chamber actually kind of acts like a ejection booth. And I'll show you exactly how that works in, I think, coming up here 2 slides from now. These guys right here, like maybe broken dynamite sticks. 7th graders, they think they look like hunched over grandmas. So this is like the hunched over back and I guess they have 3 legs, and or maybe it's a walker, I don't know, and then head. So the circles here represent different amino acids, and then this would be the transfer, the molecule that's carrying that amino acid. At the bottom, the walker legs, is the anticodon. And the anticodon is what matches complementary to the codon on the mRNA. So for example, if I have, let's just say, a codon of AUG, what would be my anticodon? Well, it's going to match complementary to it. So what pairs up with A in RNA? You'd say U. And what pairs up with U, it's A. And what pairs up with G is C. So my anticodon would be UAC. So the steps to translation: ribosome looks for a start codon. And that start codon for every single protein that's made in eukaryotes is AUG. Okay, AUG. And it codes for methionine, or MET. My 7th graders, they like to abbreviate MET as meth, but we're just going to keep it as MET. The ribosome reads one codon at a time, and a tRNA anticodon pairs with the mRNA codon. Peptide bond forms between the 2 amino acids, and the tRNA shifts to the next site that's found inside the ribosome. Then translation, these 2 steps kind of continue until a stop codon is reached, and then everything kind of falls apart. And then you end up with your amino acids that fold and become the protein. Now, all those words, gobbledygook, probably don't make any sense, but you know me, I like to model this process, okay? So I think I've done a really good job here. So let's just talk about our characters, or our players here. I got my ribosome, okay, large subunit, small subunit. I got my tRNAs carrying amino acids, and I got my messenger RNA. So the first step in translation is that our ribosome is going to find a start codon. Okay, so it fits like this. Ribosome latches on, finds a start codon. And, I didn't use letters. I used colors. And my base pairing here is blue is going to pair with green and red with gray. Okay, so for this first chamber, I have red, gray, green. Using my base pairing rule, I'm looking for gray, red, blue. So gray, red, blue is this tRNA right here. And it's going to come in, it's going to match complementary like this. And then the ribosome, it's like, okay, that matches right, moves on over to the next. And now this guy's in the next chamber. And we have an open chamber here. Using my codon, or my, base pairing of blue, green, I'm looking for green, green, and gray pairs with red. So green, green, red. So these 3 tRNAs, I'm going to move this tRNA. It slides in and matches complementary. Now, before the ribosome moves over, okay, this amino acid is going to form a bond with the amino acid that just came in. Okay, a peptide bond is formed. And then the ribosome will shift over. And notice that this chamber is like half. Well, it's like the ejection chamber, and it's going to spit this tRNA out. And as this tRNA is going to go find another amino acid that matches that anticodon. And the ribosome continues on. So I'm looking for blue, green, gray. Blue, green, gray is this amino acid, or is this tRNA. And it comes in, and we get a peptide bond that forms between yellow and brown. And we slide on over, and this guy gets ejected. Pew! Shoots it out. And then this guy comes in. All right. Peptide bond forms here. Now let's just say maybe if we reach a stop codon, okay, so everything's going to fall apart. Ribosome's going to leave, find another start codon in mRNA. mRNA, maybe it's got degraded. tRNAs leave. The peptide bond has formed. And this 4 amino acid sequence will fold into hopefully a functioning protein. Now proteins, their ingredients are amino acids, and sometimes they can be small proteins where they're only made up of maybe a couple hundred amino acids. Sometimes they're big, where they're made up of thousands of amino acids. If the protein, if there's like a mutation, this is going to lead us into section 4, but if there's like a base letter that's wrong, okay, and it codes for the wrong amino acid, well, now your protein may not function correctly, and you might end up with a genetic disorder or a disease. And, being that it's May or almost May, cystic fibrosis is one of those cases where we have these not protein channels. Here, let me just pull up a diagram. It's close. Cystic fibrosis channel mutation. Okay, so it's a mutation on the delta F508, a deletion of one amino acid at position 508 in the CFTR protein. And here we go. This is a good diagram. Slide this on over so you can see it. Okay, so we have these protein channels that line our respiratory system. Okay, and what it does is it allows chloride ions to pass on through inside to the outside, and the chloride ions keep mucus in check. So you can see we have a thin layer of mucus. Well, if you have that mutation, okay, notice that the protein is not folded correctly. This was open to allow chloride ions through, and this is closed, so chloride can't get through. And as a result, mucus builds up, and you end up with cystic fibrosis. Here's another diagram showing translation. The ribosome, the mRNA, how it's read in three nucleotides at a time, one codon at a time. Match is complementary. Here's my long growing polypeptide chain of amino acids. All right. So information tends to flow from DNA to RNA to protein. Chapter 13 was all about DNA and replication. And chapter 14 is going from DNA to RNA and then RNA to protein, which we just covered today. So the end game here is, in replication, the entire DNA molecule was copied. But in transcription, only a small segment of DNA was read, not the entire thing. A gene for maybe blue hair or for another CFTR protein channel for cystic fibrosis, whatever. Only a small part of that DNA was transcribed and then read or expressed into a protein. All right, so that does it for 14.2 ribosomes and protein synthesis.